Okay, so in talking with many of you over the past few days, a common question has emerged. Some of you are asking, what the heck do I do with this new stuff that you've been teaching? Because we're using some of the text, in particular what we're going to talk about today is in the text, but there's other stuff that we're covering in greater depth. My purpose in doing that is not to give you more stuff to try to memorize, but I want to show you how the principles that you've learned so far in 351 and 352 are the key to unlocking biology at the molecular and atomic level. And because my expertise is in the area of protein and peptide chemistry, I gave you a little bit more information there. But your goal is to be able to use information and data to make sensible hypotheses. So, as an example, with the dihedral angle stuff, do you need to memorize a bunch of graphs of amino acids? Certainly not. But I have written questions before where I said, there is a particular secondary structure that looks like this, and at this position, the dihedral angle that's preferred is like plus 60, and then 0, and then you have to, and then I give you a chart similar to the one that we went through a couple times ago, and I give you all of these individual charts for the amino acids. Do you need to memorize them? Clearly not. And then I ask you which one would fit best with the dihedral angle of plus 60 and 0, and so you look at all the data, and lo and behold, glycine tends to work, and so that's the answer. So given data, can you reason through things? If you don't know what phi and psi are, you're going to have a hard time answering that question. If you've never thought about or looked at a Ramachandran plot before, if you don't know that phi is on this axis and psi is on this axis, that's going to be difficult. Another type of question I've asked before is, given an amino acid sequence in one-letter codes only, the pH is 7.5. What is the charge on that peptide? So very similar to a question you had on your first exam, only this time I'm not showing you the side chains, and I'm probably not giving you the pKa values. You got to come with that in your brain, okay? So that's the kind of thing I want you to be able to do. This will be important to you in the future in various biochem and biology classes. You'll see protein sequences, and I want you to have in your mind a picture of sort of what functional groups are involved when somebody says this lysine amino acid or this arginine amino acid. And believe me, knowing the structures of the side chains is going to save you from some boneheaded mistakes later on in biochem. Okay, so I want to move on to carbohydrate chemistry today, but we're going to spend just a little bit more time in peptide synthesis. And this part is covered in a lot of detail in your chapter. So the sections in your study guide that refer to this are very good. The practice problems are very representative of what you might expect. We said that in order to form a particular amide bond, you've got to have protecting groups for amino acids to prevent the wrong amino group from reacting with the wrong carboxylic acid. So our protecting group for amines is BOC, which we can install with BOC anhydride. And usually in both of these cases, we need a non-nucleophilic base. I think before I've said we often use triethylamine for that purpose. But in any case, the key to recognize is just the BOC anhydride and the benzyl bromide. In any case, the BOC protects the alpha amino group, and then benzyl bromide can protect the carboxylic acid. I showed you those reactions previously, and so you can go back and review those. This first one is sort of a nucleophilic addition to carbonyls, or a nucleophilic acyl substitution, and then this second reaction is more of an SN2 reaction. Once you've protected the amino group here and the carboxylic acid here, then you can activate the carboxylic acid with DCC and then add the amine, and you'll make this lovely amide bond. Yes?
So in the book, like in this next step, you take off the two protection groups. They have a different reagent that's like carboxylic acid with HBr, and it says it takes off both of them at the same time.
Carboxylic acid with HBr.
Like carboxylic acid.
Oh, okay. Yeah, so for the last step, if you wanted to remove them both at the same time, I think they're referring to like trying to do ester hydrolysis because you can hydrolyze esters under acidic conditions. So that's fine. Go ahead.
Will we see that on a test, or will we most likely see TFA and HTBC?
So your alternative is some acid, and you probably have to heat it. That will both remove the Bach group and hydrolyze the ester, so that would be fine. The reason you'd want two different conditions is because we don't often want to stop at the dipeptide. So, for example, if we wanted to add on another amino acid, what we would first do is, depending on which end of the molecule we wanted to add it, if we wanted to add it at the C terminus, we would deprotect the C terminus of our peptide. And then you could bring in another amino acid. This one would also need to be benzyl protected. Let's just make this like leucine or something. And then you could do the same coupling step with DCC to make your tripeptide, where because you've, by strategic use of protecting groups, you are able to put in the right amino acid in the right location. And then, at the end, if you wanted to be efficient, that's a great way to do it, and that would be perfectly fine. Alternatively, if you wanted to add the next amino acid to the N-terminus, you would simply use TFA with a little bit of water, and that would give you your deprotected, your peptide with a deprotected N-terminus, whereas the C-terminus would be protected. And then you'd bring in your other amino acid. In this case, it would need to be a Bach-protected amino acid with a carboxylic acid group free. And then you could use DCC and triethylamine, and that would create your tripeptide, which I won't draw, but it would be, maybe I'll draw it anyway. So in terms of the chemistry, the chemistry is the easy part because you're just making amide bonds and doing deprotection reactions. So there's not that many reactions to keep track of. And when I go to, years ago at a peptide conference, there was a synthetic organic chemist that was introducing a new reaction that could be useful to peptide chemists. And he made some joke about how all peptide chemists know how to do is to make the amide bond, and that's really easy. And it's true, but you've got to keep track of a lot of steps. And people that do this in my lab have to pay meticulous attention to detail, because if you screw up the step and forget to deprotect the right thing, your reaction just doesn't work. Yeah?
So when you put the H3O plus in the heat, wouldn't that cause decarboxylation?
Would it cause decarboxylation? No, that only happens if you've got a beta keto acid, and we don't have that functional group here. So I think what we're just saying is if you want to use what your textbook says for one step, benzyl and Bach deprotection, that works just fine. That's just efficient. There's nothing wrong with doing it that way. There's also nothing wrong with doing it this way. Okay? All right. Questions about that? Yeah.
What's the longest peptide you usually synthesize? I know you said before.
Yeah, so what's the longest peptide that you can synthesize? Well, it's tricky, and actually there's a lot that's been done in this area. You can imagine that anytime you're doing an iterative synthesis and you're going to make something that's really, really long, you're going to have a lot of steps. Because if you think about it, for each new amino acid we add, we needed to do a deprotection and then a coupling step. And if those reactions are not greater than 99% yield, your yield's going to drop off precipitously with each step. So typically when people do, and then the other problem with solution phase synthesis, doing synthesis in solution, is the purification of your products. Imagine this reaction where you have the dipeptide and the single amino acid. You don't want to use a large excess of one reagent relative to another because you don't want a lot of one left over that you have to purify away. And as you look at this tripeptide versus your starting materials, they're probably not that different in polarity, and so purification by chromatography can be difficult. So in many cases, for solution phase peptide synthesis, people are going to stop at the sort of 10 to 15 amino acid range. Now, you can imagine that you could take various 10 and 15 amino acid segments and then link those together to make longer peptides. And back in the 60s, was it the 60s? Yeah, it was back in the late 60s, a chemist at Merck named Ralph Hirschman led a research team that used this method to synthesize a 128 amino acid protein, ribonuclease A. They didn't do it linearly. They made various segments, and then they stitched the segments together. That works okay. There are problems with that approach that are technical that we don't need to necessarily get into. But the problem of the efficiency of the coupling step, the fact that in solution you're sort of, you can't use an excess of one reagent over another to drive it to high yield, and the fact that purification can be difficult led to an important Nobel Prize winning innovation also in the 60s. And at about the same time, I think even before Hirschman finished the synthesis of ribonuclease A, another chemist beat him to it using a new method, which we're going to talk about. But other questions? Yeah?
How does this method affect folding?
How does the method affect folding? It's a good question. Also in the 60s, a chemist named Charles Anfinson showed that in many cases, the sequence of a protein provides sufficient information to direct folding. He also did this with ribonuclease A, which happens to be one of the most stable enzymes that nature comes up with and the easiest to characterize. But he basically showed you can take ribonuclease A in solution, you can boil it for a long time such that it should be denatured, and then meaning unfolded, and then you cool it back down and it adopts the right structure still. So for many proteins, the sequence alone is sufficient information to adopt the correct fold, which means it does not matter how you synthesize it. As long as you have the right sequence in the end, you're going to get the right fold. And a lot of proteins work that way. There are some proteins, particularly in eukaryotic cells, but also to some extent in bacteria, that need a little bit of assistance, not because the folded shape of the protein isn't stable, but because there are some undesired alternative conformations that can sometimes aggregate. And so cells have both bacteria and eukaryotic cells have cellular machinery that can assist protein folding. They're called chaperones, and you'll learn more about those in biochem. Okay, what else? Yeah?
Why would it be bad to have a different type of folding?
Why would it be bad to have a different type of folding? Well, there are... So there are a whole host of diseases called protein misfolding diseases. The one with which you may be most familiar is Alzheimer's, but then there's also mad cow disease and Creutzfeldt-Jakob disease. The idea in Alzheimer's is that there is a protein on the cell surface called APP, which is the amyloid precursor peptide, and it gets cleaved off by a certain protease to form a peptide called amyloid beta, and it's about 40 amino acids long. And we're not scientists are not really sure what the innate function of these molecules are, but amyloid beta can adopt a structure that's kind of like this. It's sort of a beta turn, and it's nonpolar enough that several of the beta turns stack on each other and form aggregates. And so these aggregates have been observed in the brains of Alzheimer's patients, and it's still not totally clear whether this is the cause or a symptom of the disease. But misfolded proteins, if they adopt the wrong shape, they're going to not be able to do the same function that they did previously. In mad cow disease, there's a protein called the prion protein, and the story there is just really interesting because prior to that point, it was thought to have a disease, you needed a living infectious agent like a virus or a bacterium. But the thing that infects in mad cow disease is a protein. And there's a protein called the prion protein that can exist in an innocuous, harmless shape or a pathogenic, harmful state that's misfolded. And the misfolded conformation will actually interact with other prion proteins that are folded and cause them to unfold and create aggregates that cause problems. And this is why, for example, if you go to donate blood, they ask you if you've been in the UK for more than X number of months in the past ever, because years ago there was an outbreak of mad cow disease in the UK, and people that ate beef from potentially infected cows, the prion protein from the cow could actually teach human prion proteins to misfold too. So it's a really kind of interesting story. But yeah, getting the wrong shape is often a big problem. There are other diseases called systemic amyloidoses that are related to congestive heart failure and a bunch of other things. So anyway, sorry, going off. What else? Yeah?
Have there been like ribosome mimics or like a way to synthesize protein by just using mRNA?
Yeah, is there a way to synthesize proteins using mRNA? Absolutely. So enter molecular biology, where all you have to do is feed the right piece of DNA to the bacteria, and then the bacterial ribosome will transcribe the DNA to RNA, and then the RNA will be synthesized into a protein. And that works great for a lot of proteins, and that's what my colleagues in biochemistry and in life sciences do a lot of. That's called protein expression. It's not so good for smaller peptides, which tend to get chewed up easily in biological environments. So if you want to make something that's 15, 30 amino acids long, it's better just to make it yourself chemically. The other advantage that chemists have over the ribosomal machinery is that the ribosome is generally constrained to the 20 proteinogenic amino acids, whereas chemists, as long as we can make a protected amino acid, we can put whatever we want there. And so using unnatural amino acids for interesting purposes is a big area in chemistry, and trying to find ways to get access to larger proteins with unnatural amino acids is an active area of research, and there are ways you can now incorporate one or a few unnatural amino acids into a protein through ribosomal synthesis. It's tricky though, and not everybody knows how to do it. Okay, what else? All right, so let's return to the problem of yield. If you want to make a really long sequence, you want the reaction to be really high yielding. And so in the 60s, chemist Bruce Merrifield at the Rockefeller Institute came up with an ingenious plan that revolutionized peptide chemistry and organic chemistry in general. He invented solid phase synthesis. So, and the idea grew out of polymer chemistry. So, the following polymer can be made by radical polymerization of styrene. It's called polystyrene, and it has 2 carbon units, and every other carbon has a phenyl group on it. And you've encountered polystyrene if you look at a plastic water bottle or some other kind of plastic container and it says PS on it, that stands for polystyrene. And it is literally just a large, huge polymer with these functional groups on it. And there's often cross-linking between individual polymer chains, but we don't need to talk about that. In any case, polystyrene is solid, and it's inert to a lot of chemical solvents. And so Merrifield had this really cool idea, which was, what if while I'm making styrene via radical polymerization, here's a radical initiator. You guys learned a little bit about radicals in 351. But what if I add in addition to styrene, what if I add a small amount of a benzene ring that has a functional group on it that I could do stuff with in subsequent reactions, okay? So this is the benzyl chloride version of styrene, and that's a leaving group. That's something you can do chemistry with, all right? So if you do this instead, when you make your polymer, you get the same basic structure, only occasionally, instead of just a phenyl group, you have this benzyl chloride, okay? And imagine that this polymer is basically infinitely long as far as we're concerned. And there are multiple chains, multiple sites at which you have this leaving group. And depending on how you manufacture this polymer, you can get it to form basically a bead that is big enough to see, and it's sort of a solid, and we call it a resin. I'm not sure why, but I think often you've heard of epoxy resin. The idea is a polymer that starts out as liquid and becomes solid is some kind of resin. So here is the polystyrene bead, and at various spots, both inside and outside on the surface of the bead, you have these functional groups, which are the alkyl or the benzyl chlorides. Okay, and on each bead there would be thousands and millions of these. Okay. Okay, well, who cares? Yay. And if it stopped there, I would say, right. But it didn't, because Merrifield's idea was, now, if I add an amino acid, and and some base, and I'm going to choose a non-nucleophilic base, triethylamine, that will first remove a proton from the carboxylic acid, and then the carboxylate anion can do an SN2 reaction with chloride as the leaving group. And then what do you have? Now, instead of chloro attached to your polystyrene bead, you have an amino acid. All right, now why on earth would you want to do this? Well, first, Merrifield chose the group that the carboxylic acid attached to with the idea that he was someday going to want to liberate it from the resin. Because though there are some uses for polystyrene beads that have peptides and proteins attached to them, his goal was to be able to make peptides in a more efficient way. So I will just say that this bond between benzyl carbon and oxygen is cleavable under certain conditions. But now let's talk about what you've got in the presence of this polystyrene bead that your amino acid is attached to. And remember that each of these polystyrene beads comes with tons and tons, thousands / millions of, / way more than millions of sites per polystyrene bead. Okay, so now you have an amino acid, but instead of being in solution, it's attached to this solid thing. And this thing is large enough that you can filter it out of a reaction mixture. So now imagine if I wanted to do the coupling reaction, I would first deprotect with TFA and water. That does not cleave the link between amino acid and resin, but it does remove the Bach group, right? And the idea is that after you do the deprotection reaction, you don't need to purify this, you just need to wash away the TFA. The reaction vessel looks something like this. So you have some porous filter here. It's called a frit or a filter disc, and the pores in it are small enough that individual resin beads can't get through. Whoa, that's like insane. Individual resin beads can't get through, right? So you can mix with the resin whatever reagent you need. At this stage, it's TFA or something, but and the green here is just for fun because the solutions we use actually are not green, but we're going to try to make it cooler. And then there's this outlet at the bottom, perhaps with a stopcock, which is a thing that you turn and can let the outlet be open or closed. So you do the reaction. You often have a stir bar in here to stir the resin around in the solution. But then when you're done, instead of having to isolate the product, all you do is you open up the stopcock, and that gets rid of all of the reaction, but your product stays in the syringe by physical separation. So how are we going to show this? You open up the stopcock, the undesired stuff pops out, and what's left is your resin. Instant purification. And you can even wash the resin with some innocuous solvent to get rid of the remaining TFA. But this process literally takes like 30 seconds. whereas purification of an amino acid could take hours. So it's a really fast improvement. After you do the deprotection, and then you do the deprotection to remove Bach, you drain the reaction mixture and you rinse. Then in a second step, you're going to add the next amino acid, which is going to be Bach protected. And here's whatever the side chain is. And you're going to add DCC and triethylamine. That will activate the carboxylic acid and have it react with, after step one, what you have is your polystyrene bead with your amino acid on it and a free amino group. So that when you do these two steps, you've elongated your amino acid on the resin, and now instead of having just one, you've got the second one added. Okay, and so, and again, this process involves just mixing the reagents together in this reaction vessel, stirring, excuse me, and then when the reaction is done, draining off the reaction mixture and rinsing the resin. And the fact that it's so easy to purify the remaining reaction mixture away from the peptide and the resin means that we can make reaction 2, the coupling reaction, way more efficient. We can use 3 to 5 times excess amino acid. And when you use an excess of one reagent over the other, that can drive a reaction to completion. Basically, think of it as Le Chatelier's principle. But you no longer have to worry about isolating your product from the other 4 equivalents of your amino acid starting material, because you simply wash it away and it's gone. Yeah?
So just to clarify, is in the book they were exclusively using TMOC as a protectant group and then pyridine as...
Right. So... yes and no. So the book introduced another protecting group called FMOC, and you should look at that. The study guide will take you through that. It's pretty straightforward. The reason why the book uses FMOC as a protecting group and then this amine called piperidine as a deprotecting group has to do with a fairly technical issue, which we can talk about. The linkage between resin and peptide can be cleaved, for Merrifield's original resin, can be cleaved with something called hydrofluoric acid, or HF. And if you ever take a chem safety class, they point out how nasty HF is. If you get it on you, it's not that it burns, but it does get inside into your bones, and it starts to bind calcium, and it can be lethal within days or weeks. And so if you want to handle this stuff regularly, you need to be properly trained. You need a special fume hood that you can do these reactions in. The people that do it say it's really not that bad as long as you take precautions. But the words hydrofluoric acid were sufficiently scary as to make it difficult for non-experts to adopt this approach. So people invented an alternative protecting group scheme for the amino acids, where they would use FMOC, which is like Bach, only it's base labile. That means it's stable to acid, but it comes off in base. And because you had an amino group that you could use, that you could remove under basic conditions, they were able to change the linker so that the linker could be hydrolyzed with TFA instead. Now, you couldn't use Bach chemistry with the TFA hydrolyzable linker, because then every time you went to deprotect your amino group, your peptide would pop off the resin and you'd be done. So it's a strategic choice, and FMOC and piperidine are a heck of a lot easier to deal with than hydrofluoric acid. So this is the chemistry we use in my lab. For simplicity's sake, I didn't want to spend a lot of time on it, but your study guide will take you through it and you can be aware of it. The FMOC protecting group stands for this huge, horrible behemoth attached to a carbamate. So this portion is exactly like Bach, but remember in Bach, the thing that was attached to the oxygen was a T-butyl group. Here, it's this large aromatic thing, and the way the deprotection works is piperidine, the base, removes this proton, then electrons kick down here to make a double bond, and then CO2 leaves, and then the amino group will pick up a proton from some source. So the deprotection is very much like Bach, only you need a base to do it. And so the FMOC chemistry is way more accessible, and people use it all the time now. In fact, you might imagine that this process of deprotection, coupling, iteratively would get kind of boring, and so there are now machines that can do this in an automated way. It's not actually as easy as I'm saying, but you basically program in the amino acid sequence and then it makes it for you. We do in the department have an automated peptide synthesizer. It's in my lab up on the 4th floor. But we also do a lot of synthesis of peptides by hand. And what this involves is a process where you do iterative deprotection and coupling, and you got to keep track of where you are at each step. If you don't do a deprotection, you forget to deprotect, and then you go to do a coupling reaction. Guess what? Nothing happens. That amino acid doesn't go on. And if you don't notice that you made that mistake, you continue along on your merry way until the synthesis is done, only to discover that you made the wrong thing. And everybody that joins my lab does that at least once. I've done it many times. So there's a lot of ways to mess this up. But anyway. Okay. Other questions? Yeah. How do you end up getting rid of the resin? Great. So in the final step, let's say to get your peptide off the resin, when you've done the last reaction, and the last reaction is usually removing the Bach group from the N-terminus, then you have to add whatever reagent cleaves the peptide from the resin. So for Bach chemistry and for Merrifield's original resin, this would be HF. And so you stir the resin in HF for a while, and then at this stage you can rinse out the HF, and it now has your peptide in it, whereas the resin is no longer useful to you because it no longer has your peptide. You don't throw this away. You've got to isolate your peptide after you've cleaved it from the resin. There are easy strategies for doing that, and then usually you need to do some purification. There's a method we use called high-performance liquid chromatography that's pretty easy, but we don't need to worry about that here. Okay, others? Yeah?
So in FMOC chemistry, what do you use to cleave it?
In FMOC chemistry, you use a slightly different functional group than the benzyl chloride to attach your amino acid to resin, and you cleave from the resin using TFA. One of the things I haven't mentioned that your book doesn't mention that's very important for this process, but we are going to gloss over, is that there are a lot of side chain functional groups that you can imagine would get in the way of this reaction. For example, after we remove the Bach group, we have an amino group here that's going to attack the carboxylic acid. What if this R group were lysine? What if there was an amino group on the side chain? You're not going to get any selectivity between the lysine amino group and the backbone amino group. So we need other protecting groups for the side chains, and here's the catch. They have to be cleavable under conditions that don't mess with Bach. And they also have to be stable to the peptide coupling conditions. So that's a whole other story. And ideally, you want the side chain protecting groups to be able to come off under the same conditions that remove the peptide from the resin, like HF. So for Bach chemistry, all of the side chain protecting groups fall apart in HF. For FMOC chemistry, all of the side chain protecting groups come off in TFA. It's a complication which we're totally ignoring, but if you were to come up to the lab and see what amino acids we use, you would see that they have side chain protecting groups on them, and it's to prevent undesired side reactions. All right. So that's the end of peptide synthesis. And you should know about this because it has enabled a lot of research and development. There are a number of drugs available on the market now that are peptide drugs that are synthesized via this method. There was one in the earlier days of the HIV epidemic, before there were really good antiretroviral drugs and protease inhibitors. One of the best ways to inhibit HIV infection was to use a peptide derived from one of the proteins on the viral surface, and this was like a 30 or 35 residue peptide that they made via this method, solid phase peptide synthesis. It was marketed as the drug Fusion, and it's still available, but it's not really the drug of choice anymore because there's so many better methods. But this type of chemistry has enabled a lot of research and development, and anyway. So enough about stuff I geek out about. Let's now talk about carbohydrates. And this will be in the next chapter, chapter 24. We're going to do a lot of what's in chapter 24, but we're also going to gloss over a lot of what's in 24, because a lot of what your text does in 24 is interesting to some people, but not that useful. That is, there are molecules that the chapter 24 spends quite a bit of time describing that I have never thought about in the 20 plus years I've been doing this. So if I'm not worrying about them, the likelihood that you would ever need to is like really small. So we're going to focus on the carbohydrates that you're going to encounter. So carbohydrate or sugar, we can use those interchangeably. By the way, let me just say that if you ever get into research with carbohydrates, you need to avoid the temptation to put the word sweet in your title of your paper. Everybody that does carbohydrate research wants to use that pun, and they think it's cute. And it maybe was the first 3,000 times, but it is no longer cute. Though it is true that most carbohydrates are sweet. So let me introduce you to a few, and we're going to start with the shape that the form and the shape that they adopt most often in aqueous solutions. So this is beta-D-glucopyranose. That seems horrific, but each part of that name has an important meaning. You will sometimes hear people just call this glucose, but what they generally mean when they say glucose is beta-D-glucopyranose. So let me show you the structure. It's really easy to remember the structure of beta-D-glucopyranose because it's a 6-membered ring and all the hydroxyl groups are equatorial. And then on the 5th carbon around the ring, you have a CH2 group that's also attached to an alcohol. Okay, so what do each of these, what does all of this name refer to? First, notice that this is a cyclic hemiacetal. That's the functional group there. Do you see the OH attached to the same carbon as an OR group? That is a hemiacetal. And what that tells you, once you identify the hemiacetal, the carbon that's here, the one that's bonded to two oxygens, is called the anomeric carbon for reasons we'll talk about in a little bit. Anomeric carbon. All right? And you can tell what the open chain form would look like by paying attention to what's attached to the anomeric carbon. If the anomeric carbon is attached to a carbon and a hydrogen, that means that this is a hemiacetal from an aldehyde. Glucose is from, in this cyclic hemiacetal form, also has a form that is an aldehyde. And sometimes people will call glucose an aldose, which means it's a carbohydrate where carbon 1 is an aldehyde. 1, 2, 3, 4, 5, and 6. Okay? So seeing the hemiacetal carbon is really important in carbohydrate chemistry, and being able to recognize, oh, in the open chain form, that anomeric carbon that's the hemiacetal would actually be an aldehyde is really important. So I'm drawing the open chain form for you right here in the same overall shape, just we now have the aldehyde here and the OH group on carbon 5. People get confused whenever you're making rings. Your mental SpongeBobs go all over the place and the whole thing lights on fire. So you make a 6-membered ring, but it's between the OH group on carbon 5 and the aldehyde on carbon 1. Now the mechanism for that, presumably you should be really good at. If you're not, you need to go back to the early chapters this semester and look at acetal and hemiacetal formation. Okay, so let's talk about each element of the name. The ose refers to the fact that this molecule is a carbohydrate. So if you see ose at the end of a name, it means it's a carbohydrate which has the general formula CnH2ON, or a hydrate of carbon. Fructose, sucrose, maltose, dextrose, all of these things are carbohydrates. Next, how about the pyran part? The pyran part refers to the 6-membered ring form of the sugar. So if something is a pyranose, that means it is a 6-membered ring. Pyran is a 6-membered ring with an oxygen in it. So, but in in this context, pyranose means that this sugar is it forms a 6-membered ring with an oxygen in it. Okay, gluco refers to the stereochemistry of carbons 2, 3, and 4. Why would you only want to talk about the stereochemistry of carbons 2, 3, and 4? Well, I don't know. I didn't invent the rules, but the beta and the D refer to the other stereochemistries. So the fact that the name gluco tells you that the OH group on carbon 2 is down, that the OH group on carbon 3 is up, and that the OH group on carbon 4 is down. If we switch one of those, we would have a different sugar. For example, if instead we had down, up, up, that would be galactose. And we would use galactopyranose instead of glucopyranose. Okay, so we'll continue with that next time. We'll learn more about these names and more about carbohydrate structure.